This protocol allows a researcher to determine whether there are differences in the energetic balance of mice, which can help identifying the physiological processes responsible for a change in body weight. It can also be used to determine the capacity of brown fat to expend energy. This technique quantifies the oxygen consumed and the CO2 produced by an individual mouse in a controlled environment. It allows users to calculate the energy expenditure in this individual mouse and determine whether different treatments or genetic manipulations can change energy expenditure.
To begin, open the software controlling the enclosure and the airflow, and let the software test the computer's communication with the equipment. Once communication is established, click File, then Open Experiment Configuration, and select the experiment configuration pre-designed by the vendor or set up from a previous assay. Next, click on Experiment, then click on Properties to open the Experiment Properties window. In the Properties window, set up the parameters of the environmental enclosure, including the ambient temperature and 12-hour light cycles. Then click on Experiment, followed by Setup, to open the Experiment Setup window, where the parameters of each metabolic cage are defined. Next, add a pre-weighed amount of food to the feeders in the metabolic cages housing 8-week-old female mice. Also, place the water bottles and check that the bottles are